title co-regulation of human PCNA and PCNA antisense RNA1. In my talk, Dante's introduction was the introduction, and I will say some facts about PCNA and how PCNA functions in vivo, and classes of non-coding RNA, and some examples how the different genes are regulated by the antisense RNA. And in the results part, I will talk how we can form the PCNA antisense RNA gene and the co-regulation of PCNA and antisense RNA at the translational level and the PCNA gene regulation at transmission level. And finally, discussion and conclusion. So first, introduction of PCNA. What is PCNA? PCNA means the proliferating cell nuclear antigen, and it is also called the master of the genome. Molecular weight of this protein is 29 kilodaltons, and this is the cofactor of DNA polymerase delta. So it is necessary for DNA replication, cell cycle progression, and cellular proliferation. The expression of the PCNA is lowest in the serum starch cells, and it's highest after 18 hours of serum stimulation. So it means its expression is highest after the S phase of the cell cycle. And two isoforms of PCNA have been identified so far in breast cancer cells. And the PCNA is so important that if we knock down the PCNA, the DNA replication stops and ultimately cells die. And then one interesting feature of this gene is the extensive sequence similarities between introns and exons and among introns. This is the schematic outline of DNA replication. I said in the start that this is called the master of the genome because of its strange structure. As we know, when the DNA is replicated, so there are two strands, one is the leading strand, one is the lagging strand. So the nascent double-strand DNA, which is newly made, it has to pass to this ringmaster. If we disturb this ringmaster, then there is no efficient DNA replication, and ultimately cells will die. So it means it's very important and the initial basic step in the DNA replication and cell cycle. So as my talk include the PCNA regulation by non-coding RNA, so I would like to classify non-coding RNAs. We have two main types, housekeeping non-coding RNAs and the regulatory non-coding RNAs. In the housekeeping non-coding RNAs, we have ribosomal RNA, transfer RNA, small nuclear RNA, and small nuclear RNA. And these all RNAs expressed constitutively. In regulatory non-coding RNA, we have further two types, the small non-coding RNA and long non-coding RNAs. In small non-coding RNA, we have microRNA and small interfering RNA, pRNA, etc. But mostly the long non-coding RNA, which is very emerging field, very important in the gene regulation. We have two types. One is the sense non-coding RNA, and one is the antisense non-coding RNA. The sense non-coding RNA means when the non-coding RNA is expressing from the same strand of the DNA from which the respective gene is expressing. But in the case of antisense non-coding RNA, its expression is from the opposite strand of the double strand of the DNA. So means the antisense RNA is expressing from the plus strand of the DNA, and the sense gene should express from the negative strand of the double strand DNA. So my main talk is the regulation of the gene by antisense RNA. So basis on the classification of the antisense RNA, we have 5 types. In the first type, the antisense RNA and the sense RNA gene, they overlap each other in the head-to-head manner. And the second type, both the sense and the antisense RNA overlap each other in the head-to-head manner but non-coding region. In another type, the sense and the antisense RNA overlap each other but in the 3' end, and also in the 3' end but in the non-overlapping region. And the last type, the antisense RNA is fully overlapping the sense gene. And the talk which I'm going to present is about this type of sense antisense RNA overlapping. In this case, the antisense RNA is fully overlapping the PCNA gene. So first I want to show a couple of examples to see how the antisense RNA is regulated in the gene regulation. So this is the one paper which was published in Nature in 2008. In this paper, as we know, the P15 gene is also a very important cell cycle regulator gene. This P15 gene has one antisense RNA, and that antisense RNA is called P15 antisense RNase. So this means the antisense RNA is transcribing from the opposite strand to this gene. So in this case, what they did, and another thing, the P15 gene expression is high in the normal cells, but the antisense RNA gene expression is high in the cancer cell, just like in lymphoblastic leukemia and acute myeloid leukemia patients. So they made three constructs. In the one construct, it has the P15 gene, and the second construct, it has the antisense RNA, but then mutated, so it's just a control. And the third construct, it has the antisense RNA. And through phage and GFP expression, after 3 days of the expression of this vector, we can see the GFP expression is 5.8% and 6.6%, because they took these two as a control. And in the third, these cells, they express the antisense RNA also. So we can see the expression of the GFP significantly decreased compared to these two transfected cells. So in this case, they grow the cells for further 3 weeks to see more clear results. So we can see here that these two transfected cells, the GFP expression is very high, which is 51 and about 60%. But in the third construct, which is the antisense RNA, the GFP expression is very low. So it means the antisense RNA is affecting the gene's expression in the cancer cells. So later they found that, as we know, the H3K4, which is the active gene mark, and it helps in the active gene transcription, and H3K9, which is the repressive gene mark. So they found that those cells in which they have expressed the antisense RNA, the H3K4 mark decreased compared to the control cells. But in these cells, the H3K9 mark increased. So it means these cells, the antisense RNA has affected the gene expression through heterochromatin formation, and this P15 genes cannot express in these cancer cells. And here's one other example, how the antisense RNA regulating the sense gene. This is also one cell cycle regulator gene, the P21. So in this case, it has the antisense RNA gene. The expression of both the sense and the antisense level is a normal level in the normal cells. But after the knockdown of the antisense RNA, the expression of the sense genes increases significantly. But interestingly, along with this expression, first the H3K27 trimethylation mark, which is the repressor mark, it was present at the promoter of this P21 gene. But after the knockdown of the antisense RNA, not only this H3K27 trimethylation disappeared, but it also dislodged the AGO1 protein, which is known to be help in the sRNA knockdown of the gene. But in the opposite case, when in this the author knocked down the AGO this gene sense gene, the expression of the antisense RNA increased. But interestingly, not only the gene expression increased, again the H3K27 trimethylation mark reappeared along with the AGO1 protein. So it means the antisense RNA also affect the gene expression through chromatin modification. So based on these results, we found that there is one antisense RNA gene which is present in the overlapping the first exon of the PCNA. The PCNA gene has 6 exons and 5 introns, and it was already suggested that there may be some promoter in the first intron of this gene, and it was suggested that this promoter may be present in this location because there were some transcription factors which were binding here. So the size of this antisense RNA, according to the UCSC genome browser, it is 384 base pair, and the size of the first exon is 424 base pair. So first, we confirm the expression of this antisense RNA gene in 2 cells, in the HEC293 and HeLa cells. So we ligated the pi prime adapter in both the RNA and the HEC293 and the HeLa cells, but we couldn't see the clear band, so we did the nested PCR and then confirmed the expression of this gene by TA cloning and sequencing. So as it was suggested that maybe if the expression is there, so it has some promoter which should be present in the first intron. So we cloned the first intron of this antisense RNA, so it means the sequence is from the first intron of the pCNA. Although the promoter of the pCNA is very strong, but there's also some promoter activity in the first intron of the pCNA. So it means the antisense RNA is regulated by this promoter. So next, if the antisense RNA is present there, so it is also very important because pCNA is recorded in the cell cycle propagation and cell regulation. So we knock down this antisense RNA by siRNA. So it was expected that the siRNA decreased the antisense RNA gene expression, but along with this decrease in the expression, the pCNA expression also decreased both in the HeLa and the HEC293 cell. So this shows that the antisense RNA is regulating the pCNA gene expression. Is both the pCNA and antisense RNA are overlapping each other, so if the antisense RNA is regulating the pCNA, then how about the pCNA, whether it can regulate the antisense RNA or not? So for this purpose, we targeted the pCNA by siRNA. In this case, again, in the HeLa cells and HEC293 cells, the pCNA expression was decreased as expected, but along with this pCNA, the antisense RNA expression was also decreased, both in the HeLa and the HEC293 cells. So this shows that the pCNA, although the pCNA antisense RNA regulate the pCNA gene, it has also the ability to regulate the antisense RNA gene. So after this, we confirmed our result by Western blot to see whether this antisense RNA can regulate the pCNA at translational level. So here we can see after the knockdown of the antisense RNA, the protein level also decreased in the HeLa cells as well as in the HEC293 cells. So in this figure we targeted the pCNA. So as was expected, the pCNA expression also decreased both in the HeLa and the HEC293 cells. So after this, it was classified that in the case of the sense and antisense RNA gene regulation, there are two types of regulation. One is called the discordant regulation and one is called the concordant regulation. In the discordant regulation, if the antisense gene is knocked down, then the sense transcript is increased. But in the case of the concordant regulation, when the antisense gene is knocked down or decreased, the sense transcript also decreased. And in this paper, they have also given a few examples which shows that the phenomena of concordant and discordant regulation is already present in different genes, both in mouse and in human. So this was one paper we published last year. In this case, this pCNA has also one antisense RNA, although the size is different both in the human and the mouse, and the location is also different. In this case, this antisense RNA is present in the promoter region, and also the same promoter, both the pCNA and antisense RNA are sharing the same promoter. So in this case, the result was different from the human cells. In the normal cells, mesenchymal cells and the embryonic fibroblast cells, after the knockdown of the antisense RNA, the pCNA expression was decreased. But in the case of the cancer cells, melanoma cells and lung carcinoma cells, after the knockdown of the antisense RNA, it didn't affect the pCNA. So it means that some another mechanism is present in normal cells, and that mechanism are different in normal cells and cancer cells for the regulation of the pCNA gene. And this is also a very nice example to show how the antisense RNA is regulating the sense RNA gene. The beta secretase gene is very important in the Alzheimer's disease. In this case, it has the antisense RNA, which is overlapping the one exonic region in the sense strand. So in this paper, they have shown that through RNase protection assay, if both the sense and antisense are not present, then the another is decreased. For example, they use probes and RT-PCR. In the case of absence of RNAs, both we can see the expression is there, but after the treatment of the RNAs, there is expression of the overlapping probes but no expression of the non-overlapping region. So it means both the sense and antisense RNA are making the double strand. And in this case, they have knocked down the antisense RNA gene. So after the knockdown, the sense RNA expression decreased compared to the control cells. And after the overexpression of the antisense RNA, the expression of the sense, the beta secretase gene, increased. So this also shows that the sense and antisense RNA can regulate each other by making double strand RNA. So this is the conclusion of my talk. After the knockdown of the antisense RNA, the pCNA gene expression is decreased. And similarly, after the knockdown of the pCNA, the antisense RNA expression is decreased. So from the work so far, we concluded that the pCNA and antisense RNA co-regulate each other in the concordant manner. So these are a few questions which I think are very interesting to me. If the sense and the antisense RNA are transcribed by RNA polymerase II, then how both are not colliding with each other because they are going in the opposite direction? And another, what is the exact mechanism? Because the double strand may not be the only mechanism to regulate each other. And as the pCNA is very important in the cell cycle, so could we exploit the antisense RNA for the regulation of cell cycle, especially in cancer? And I'm thankful to my Professor Zayang Rose, and other lab members for their contribution.
It's a nice talk. Any questions?
So, do you know the radical label of antisense? Oh, radical label. How about they are? Do they 1-to-1 or 3-to-1?
Actually, I don't know exactly, but the expression.
But that's a very important question. So, because you use PCR, you cannot tell that. Very difficult to tell. Because one. So, I think worthwhile to doing that, try to quantify differences.
Yeah, but at least the antisense RNA expression is lower than pCNA.
Oh, yeah. That's assumption. So.
Yeah.
Okay. The second issue, what is the mechanism in the pCNA situation? Is the transcription initiation interference? Or
No, actually, there are a few possibilities that the antisense RNA is used in the pCNA. First, it may make a double strand. It may make a double strand RNA, so it is helping to protect the pCNA from degradation. Or there maybe it may help in its chromatic modification. So that it is the next work which I'm planning to do. But at this time, I don't know what is the exact mechanism. Because I give a few examples in that case. In overlapping, one is making a double strand, and the other is some chromatic modification. So we don't know the exact mechanism.
another issue is have you tried to mapping the initiation, the 5' end of antisense transcript?
Actually, I
and
tried.
3' end, because that's a very important question.
Yeah, actually,
If you have that information, you may know the mechanism much better.
Yeah, I tried that, but I couldn't succeed so far on that.
Okay, I have a question. So I saw in your study, most of the time you used siRNA. Did you use the overexpression of this non-codamine and see the effect on the pCNA? I don't remember I see that. When you overexpress this pCNA antisense.
No, I didn't do expand overexpression so far. Because in the case of the antisense RNA, we don't know the exact size. So I cannot overexpress that.
Yeah, you have to do the 5' and 3' raise to determine the exact end. I remember in last year there was a Nature paper that reported this a category of antisense non-RNA, which represses the expression of the mRNA because to compete for the ribosome binding sites. So, because they have a detailed mechanism, that's why they're in nature. And I mean, it's a very interesting phenomenon, and it's worth studying the further mechanisms.
Yeah, but I saw in one paper that about in mouse about 70% of the genes have the antisense RNA, although we don't know all of them. So it is maybe different mechanisms for regulation of different genes.
Yeah, that's right. But in the nature paper, they identify certain fragments which are common to that category of monoclonal. Thank you.
Thank you.